Welcome to tonight's AMDA webinar titled Genzyme Next Generation Pompe Treatment Clinical Development Overview. Your guest speakers for this evening's program are Edward Kaye, MD, and Alison McVie-Wylie, PhD. Edward M. Kaye, MD, is currently Group V.P. and Therapeutic Area Head in Clinical Research at Genzyme Corporation, where he supervises the clinical research in the lysosomal storage disease programs and in the genetic neurological disorders. Alison McVie-Wylie is the Scientific Director of the Pharmacology and Toxicology Group at Genzyme Corporation. Alison manages a group of scientists who design, execute, interpret, and report studies to support preclinical drug development of novel biological therapeutics. Alison was the lead scientist responsible for the preclinical development and licensure of Myozyme. Operator assistance is available at any time during this conference by pressing £0. A question and answer session will follow the presentation. You may send chat questions at any time during the presentation using the chat window indicated by a speech bubble icon located to the right of the presentation. A recording of this webinar will be posted onto the AMDA webinar for a limited period of time. I will now turn the call over to our moderator, Kathleen Coolidge from Genzyme Patient Advocacy. Please begin.
Hi, everyone. Thanks very much, Leo. My name is Kathleen Coolidge, and I work at Genzyme. I'm in the Patient Advocacy Group, and I just wanted to welcome everyone to this evening's webinar. In particular, I'd like to thank the Acid Maltase Deficiency Association, Tiffany House, and Marcia Zimmerman for initiating this request to Genzyme to provide an update to the Pompe community about this research. We particularly appreciate all the effort that has gone on behalf of the patients to reach out to them by Marcia and Tiffany, so thank you very much. We're eager to get started, so at this point I would just like to turn it over to Dr. Ed Kaye.
Good evening, everyone, and thanks for joining us. I'm going to, this will be a joint presentation. I will talk a little bit about our research and development and what we've done so far in Pompe disease, and then Alison will talk about some of the current research that's ongoing for neoGAA, our next generation material and drug for Pompe disease, and then I'll round up and talk just a little bit about an overview, and then we'll have time for the questions and answers. So what we'll talk about today then is really the history of the Pompe development program, which many of you know has been beginning since 1998. Talk a little bit, what Alison will talk about is some of the advances through research that we've discovered, and also then what is our overall plan for research and development for the next generation Myozyme. So the history is quite long and really starts using the transgenic material back in 1998. And a lot of work, of course, was done in the Netherlands with Arnold Reuser and Hans van der Ploeg. And as we've gone through, there's been a number of really pivotal experiments and studies that have been done that allowed therapy to be developed and brought to the market. One of the concerns, of course, is in the early stage of the program, we in fact had three different products, three different recombinant enzymes for treatment of Pompe disease. And in order to really resolve that issue, and some of you probably saw it in the movie Extraordinary Measures, there really was an experiment called the mother of all experiments in which the three different forms of the recombinant protein were tested. And at the end, the current formula was the one that worked the best, and that was chosen to go into the market. Now, once we had a drug, then we had to test it in the various patient populations. So the first and the neediest population were the infants, and that really started and completed started in 2003. And then as part of the development plan, we looked at the natural history in the late onset population. We looked at some of the most severe, also late onset population, and then we did the full study in the late onset, the LOTS trial itself, and that completed and finally culminated in approval for Myozyme, both in Europe and the U.S. in 2006, and just recently in June of 2010, Lumizyme, which is the 4,000-liter product marketed in the United States, was approved by the Food and Drug Administration. So it's been a long process, and I think the journey is not over, and there's a lot of work that we need to do and work that has to be done by investigators and patients alike to try to really understand, can we do better in the treatment of Pompe disease? So right now, the Myozyme or Lumizyme, depending on where you're located, is really throughout the globe, and there were over 1,400 patients who are on therapy, and commercial, some clinical, and also some charitable donations. And there are 900 patients in the Pompe registry, so which really began in 2003. So it's grown a lot, and I think as we have more patients who are experienced on the drug, then we realize what are some of the needs, and also how can we make it better. So just to remind people of the process, and typically in drug development, you begin with a smaller scale during the early phase of the drug development program. So we started with the 160-liter process, and this was used for the infantile study. And then as a development program, obviously to meet the world supply, it had to go to a 2,000-liter. Currently, that's no longer in use because we now have a 4,000-liter material, and this is used both in Europe and the U.S. and Japan, and this is known as Myozyme outside of the U.S., and the 4,000-liter material is known as Lumizyme in the United States. But this is the primary drug that is used to supply most of the patients throughout the world. One of the complicated aspects about Pompe disease is that although we know the gene and we understand the enzymatic defect, which is a deficiency of alpha-glucosidase A, the phenotype can be quite different. So there are, even with the same genetic mutation, you can have some patients that have the severe infantile form that present typically within the first few months of life and are severely affected with a cardiomegaly, severe respiratory problems, and profound muscle weakness and hypotonia that usually leads to death within the first year of life. But in addition, the most common form of the disease is really the late onset form. And these are adults and children that typically begin after 1 year of age and don't have the cardiac involvement, but still have a severe disease because it results in severe disability, proximal muscle weakness, problems with walking, using the arms, often leading to the use of a wheelchair, problems with respiration because the diaphragm is affected and leads to problems with sleeping at night, and eventually problems which may require a ventilatory assistance, and eventually leads to a premature death. So both are severe forms of the disease. One starts very early and is very aggressive. The other is more indolent. So a lot of work has been done to try to look at the entire population of Pompe disease. And remember, early, because of the acute need for infants, a lot of the work was done in the infants. And so we have the original, what was called the pivotal trial, which was 1602, and that was recorded and published by Dr. Kishnani. But also we have the older infants who were between 6 months and 3 years. Dr. Nicolini had published that. And then we, of course, have all of the kind of the intermediate stage. So we have the severe infantile or the very severe late onset patients. But again, the majority of the patients are still walking, ambulatory, and without the use of a ventilator. And that's the population that was studied and recently reported in the New England Journal, and Dr. Vanderplug was the author for that. That study, which was the basis for approval in Europe, the United States, and Japan, was also recapitulated using other, in Germany, with Strathholt recently. They had very similar findings to what was found in the pivotal LOTS trial, and also Dr. Angelini in Italy, in 11 patients, found similar responses to the therapy. So we do understand the disease somewhat, and we understand a little bit of the biology, but we're still learning a lot as we go through. One of the important aspects of a disease that we've certainly found in all of our drug development programs is that it's not enough simply to provide the therapy. It really is, it does take a village to treat a patient with Pompe disease, and it's not just simply supplying the drug, but it's supplying all of the other care that the patient needs. So this is an example, and this was in a publication from Dr. Kishnani, really talking about what's the appropriate therapy and the care and the guidance that should be given to physicians taking care of this. Well, one of which is you need to have a number of specialists. So the children often will have certainly have the pediatrician who is the captain, so to speak, but will have a neurologist or a muscle expert that works with them, a pulmonologist, frequently a cardiologist for the infants. And then in addition to the physicians in the treatment, there's physical therapy, there's respiratory therapy, there's genetic counselors that are involved in the care. And there's some basics such as nutrition and exercise, which are really critical for the well-being of a patient with Pompe disease. So all of these are necessary in order to have a successful outcome. And there's a lot to be learned. And for patients with Pompe disease, obviously, we need to understand the disease itself, but we also have to be able to have instruments that can monitor whether a patient is doing better or getting worse. And we have to realize what the sensitivity of those instruments are, and then eventually how does that inform us in developing a new treatment. So there are a lot of research efforts that do continue, and some of the work that Genzyme has been involved with and working with others, a very important breakthrough that helped us with the early Pompe work was to make a diagnosis earlier for these patients. And for those of you who recall, Pompe disease is complicated because it initially was not a simple blood test. There was another enzyme that would interfere and give people a false, a negative study. It appeared as if they did not have the disease, while in fact they did. So in order to get around that, Mr. Tremoles had done some breakthrough work on putting blood on a filter paper card, which allowed people to send it in, to mail it into a laboratory, and then he used an inhibitor for the interfering enzyme and was able to show that you can in fact diagnose Pompe disease in blood, even simply in a filter paper card. So that shortened the time it took for patients to be diagnosed, and that is a critical piece of information, especially for a very young infant who needs treatment very quickly. Also things such as a high protein and low carbohydrate diet and exercise. Dr. Alf Slonim in New York has worked on this for years, and now he has done research, and other people are looking at research to see what is really the response for exercise. How is someone who is on therapy, how do they get better if they have exercise in addition to enzyme replacement therapy? Dr. Kishnani has worked on immune modulation, especially for patients with antibodies and in response to high antibody titers, which might interfere with the enzyme. How do you reduce that? How do you get a better outcome? Dr. Nina Rabin at the NIH has been working on autophagy, and if there's a way to modulate that and potentially improve the outcome. And also there's ongoing work right now at the University of Florida with Dr. Barry Burns on gene therapy, and a lot of work on targeting to the muscle lysosome to get rid of glycogen. So what would we like if we could have the ideal next generation therapy? And often when we're in the development process, we get together and try to say, well, what would we like the drug to do? And this helps us guide our overall development process. Well, one of the things that came out in our discussions with Genzyme and talking with patients and also with investigators, people would like really a greater and more consistent efficacy along many of the stages of the disease process. So can everyone respond in a better way to a drug? And obviously that would be an ideal type of therapy. We also would like the drug to have at least a similar safety record and, if possible, an improved safety record in regards to infusion-associated reactions or antibodies. And finally, if we could have everything that we wanted, we would love to have a more conveniently delivered therapy, whether it would be an intravenous therapy that could be given less often, or even something such as an oral therapy if that was possible. So I think the first step in the whole drug development process is really trying to understand the biology of the disease. And this cartoon talks a little bit about what we know, and I have to emphasize that this is a very simple schematic in which we've simplified the process. I just returned from a Gordon Research Conference last week where we spent 4 days talking about the biology of the lysosome. And one of the things I took home from that is that there are probably close to 150 genes that are involved in just the membrane of the lysosome and tells it when to fuse with another lysosome or with an autophagosome. And also lysosomes are involved in repair of membranes of cells. So the lysosomes do a lot more than simply process or degrade material. It's a very active organelle that has a lot of functions in the cell. But what we do know is that when glycogen is in the cell, it needs to be broken down to be recycled. So one of the pathways in the Pompe disease is the only glycogen storage disease that involves a lysosomal enzyme. All the other enzymes are in the cytoplasm, which is outside of the nucleus but not in an organelle. And so we know that the glycogen needs to get into what's known as an autophagosome, and that's the part of the organelle of the cell that digests it. And then eventually there are triggers and various proteins and genes that allow it to fuse with a lysosome where the enzyme is located. Then it's broken down, and then that glucose can be excreted out of the cell into the circulation. We know when that system breaks down, when you have a deficiency of the enzyme, the glycogen accumulates in virtually all of the tissues, but especially in skeletal and cardiac and diaphragmatic muscle. So one important thing, if we're developing the therapy, especially if it's a recombinant enzyme replacement therapy, we need to be able to deliver the new therapy and get it into the target cells. So it's not going to do the patient any good if it doesn't get into the muscle or into the heart or into the diaphragm. So the body has a way of transporting this recombinant protein from one cell to another. So the important aspect of this transfer is known as the mannose-6-phosphate receptor. So that's a tag on the outside of the cell that allows the enzyme that's in the circulation to link up, almost like a space station where the rocket links up onto the space station. And then it has to have a way of getting in through that membrane, and there's a mechanism in place that opens up the pores and allows the enzyme to enter. And then in order for it to work, it has to find its way into the lysosome. So the first organelle is the early endosome, and then we think it becomes a late endosome, and then eventually it forms into the lysosome. And in the lysosome, which has a very acidic environment, that's where the enzyme does its job and break it down. And so anything that interferes with that process causes the accumulation of glycogen. So one of the basics of the disease that we need to think about is, even though we understand the gene defect and we understand the enzyme defect, we still don't really understand what's the cause of the pathology. What causes people to have weak muscles? Initially we thought, well, it was just a lot of glycogen and it would gum up the works, so to speak, but it's become much more complicated, and we're still trying to understand the biology so that we can intervene at the right time. So we know that early in the process, these lysosomes fill up, but the glycogen stays within the lysosome, and if you stay in the cell, you can actually see it, and you can see the glycogen within these swollen lysosomes. Over time, however, what occurs is the lysosomes get larger and larger. They're no longer able to function, and they break down. And so that glycogen that was in the lysosome now is spread in the cytoplasm. And remember what I said, in order for this enzyme to work, it has to work within the lysosome. So now we have a lot of the glycogen that probably can't be broken down because they're not in the right organelle for the recombinant enzyme to work. So over time, this accumulation increases. You end up having a lot of spillage of the glycogen out of the lysosome into the cytoplasm, and eventually it does damage the muscle. We get fibrosis. You get a muscle that's no longer able to contract. And at that point, we think that it probably is too late to try to intervene with therapy because the muscle is too severely damaged to recover. So we don't have an easy way to restore that muscle or to put in new stem cells to be able to have the muscle come back to normal. So we have to be able to do a better job at diagnosing the patients earlier, intervening when they have the best opportunity to respond and have a more normally functioning muscle. Now I'm going to turn it over to Alison.
Good evening. As Ed has mentioned, the mannose-6-phosphate receptor is extremely important in regards to cellular uptake and targeting of the recombinant enzyme to the lysosome. We know that our current treatment is 110 kilodalton in size. It has 7 glycosylation sites, which is where sugar moieties like mannose-6-phosphate is attached to the protein backbone of the enzyme. However, we do know that we have a limited number of mannose-6-phosphate on our recombinant enzyme, and we have even less of something called the bis mannose-6-phosphate. And the bis mannose-6-phosphate is simply just a glycosylation molecule or a sugar molecule that contains 2 mannose-6-phosphate. And it has been shown in the literature that the affinity of the bis mannose-6-phosphate is much higher for the mannose-6-phosphate receptor compared to the affinity of mannose-6-phosphate. So our hypothesis was to create a molecule in which the number of bis M6P on the sugar sites of the recombinant enzyme was significantly increased, and we hypothesized that then that would lead to greater glycogen depletion and potentially better clinical outcome. So our strategy in which to generate a molecule with an increased number of bis M6P was to chemically conjugate a synthetic glycosylation or a synthetic glycan that contains this bis M6P and make a molecule that we could then investigate ultimately human safety and efficacy and compare that to the recombinant enzyme that we have available today. So these cartoons highlight schematically our strategy to do this. So on the left-hand panel, I hope you can see the arrow. This is a recombinant enzyme that we know as Myozyme or Myozyme. The green portions of this 3D picture of the molecule are the glycosylation sites. And this small red fraction at the end is the mannose-6-phosphate that we know is contained in the recombinant enzyme. So our strategy is to synthetically conjugate the synthetic glycan that's shown here. The blue circles depict a synthetic linker that we have that we can chemically then conjugate the glycan on with the 2 mannose-6-phosphate containing sugars or the bis mannose-6-phosphate that we have now decorated this recombinant enzyme with. So once we had generated that candidate in the research group, we then began the pathway through research and development. And in order to optimize the candidate that we had chosen to move forward as a second-generation approach, we did a multitude of in vitro or analytical testing, and that includes looking at the catalytic function or the enzymatic activity of our new molecule. We also have the ability to fully characterize the glycosylation pattern so that we know exactly what we've added onto the molecules. We can look at cellular uptake, which is looking at the uptake of the molecule in a cell culture system where we could look at fibroblast cells or skin cells. We can look at muscle cells and take a look at its uptake there. And we can also look to see how this new enzyme is processed within the cells so that we can understand that. We also do some in vivo testing using the Pompe mouse model that was developed by Nina Raben at the NIH. And by administering the enzyme to the Pompe mouse model, we can look at glycogen clearance, we can look at tissue distribution of the enzyme, compare that to Myozyme or Lumazyme. We can look at the pathology and the impact of our new enzyme on the pathology of the disease. We can also look at a functional response, and later I'll speak to that. And because we have a battery of functional tests that we can put these animals through so that we can test their muscle function. And of course, we also look at safety. And then we would move on to clinical testing, or we move on to clinical testing, which, as you all know, will involve, the administration of this to patients in a clinical trial. So one of the first experiments that we did when we were evaluating this approach was to look at various structures of the different glycans that could potentially be attached to the recombinant molecule. So if we look at these cartoons, these show 4 different structures that we evaluated so that we could determine really what was the most optimal structure that we could put onto recombinant GAA to make the most potent enzyme in terms of glycogen clearance. So these cartoons, the blue shows the linker and the circles are the sugar molecules. Again, the red circle shows the mannose-6-phosphate. So we looked at a simple structure that only contained 1 mannose-6-phosphate. We looked at another molecule that contained 4 sugar molecules in total with 2 mannose-6-phosphates. And we looked at 2 other molecules that both contained 6 sugar molecules. However, they were in a slightly different orientation, which can really impact the binding of that sugar moiety to the mannose-6-phosphate receptor. We then generated all of these molecules, conjugated them to enzyme, and administered them to the Pompe mice. The results of the experiment is showing here in terms of glycogen clearance. And you can see that this molecule, which is termed here SAM6-beta, which at the time was an internal name for these molecules, was the one that showed to be most potent in vivo in the animal studies in terms of clearing the most glycogen from the skeletal muscles. We also did another experiment where we looked at the number of glycans we had to conjugate onto the molecule to see if we could see what the most optimal number of glycans would be. And in this representative experiment, you can see that we did show that we saw the maximum efficiency required at least 3 glycans. And you can see that that's shown here when we look at 3 glycans of this molecule. And this is our new neoGAA molecule, dosed at either 4 mg/kg or 20 mg/kg. We can see that that is similar to 20 and 100 mg/kg of recombinant enzyme. And it also looks similar to a molecule we made with 8 glycans. So we've very heavily decorated with the new glycan. As mentioned, we have done substantial in vitro and analytical analysis on these molecules. You can see from this experiment where we looked at GAA uptake into myoblast cells or muscle cells in culture, you can see that we have much more efficient uptake into the muscle cells for the neoGAA compared to the GAA itself. And we also have another experiment called a Beer-Core experiment, in which we also looked at the affinity using a different method, and we saw that it was at least 10-fold higher based upon the results of that study. These graphs show our results of evaluating our neoGAA candidate. So this is the SAM6 molecule, the optimal molecule that we put into the Pompe mice. You can see that we put it in as a dose of 4, 12, and 20 of the neoGAA versus 20, 60, and 100 mg/kg of GAA. And you can see that at 1/5 of the dose of neoGAA, we saw similar clearance as we did with the recombinant human GAA. And you can see in these histology slides in which these have been stained for glycogen the glycogen is stained the purple color, you can see significantly less glycogen in the tissues of the animals given neoGAA at 20 mg/kg versus that in the tissues of mice given recombinant GAA, Myozyme or Lumazyme at 20 mg/kg. And we saw that both in the heart and the skeletal muscles. And finally, with our preclinical experiments, we also did some functional testing of muscle strength in the Pompe mouse. And we have two tests that we've shown here that we have the ability to do. The first looks at muscle coordination, and that's called a rocking rotor rod. And that's where we put the mouse on a rod that spins very quickly in one direction, then it stops, it reverses and spins in the other direction. So the mouse has to be very coordinated to be able to stop when the rod stops, turn around, and start running the other way. And this is a great test to look at muscle coordination as well as muscle strength, because they have to be strong enough to stay on. The other test that we use is called a wire hang, in which the mouse is allowed to hold on to a wire rack, and we can test its muscle strength in terms of how long that mouse can hang on for. And you can see that the results of this experiment show that these Pompe mouse, the Pompe mouse model, over time in the vehicle groups, so these are the untreated mice, you can see that this experiment was started -1. The animals are 4.5 months of age, and you can see even at that point that they have significant deficiencies in their muscle coordination as well as in the wire hang or the muscle strength, and in the animals that are untreated, this declines over time. You can see in the pink we have what a wild-type animal would do in the same testing. And you can see that 20 milligrams of the neoGAA maintains muscle strength in the wire hang test, and it also starts to improve muscle coordination over time and over treatment up through 8 months of treatment. We see a similar response with Myozyme. However, we do have to note that the Myozyme that was given in this experiment was at 100 mg/kg. So again, a 5-fold higher dose than what we used for neoGAA. So with that, I'm going to pass back to Ed, who's going to talk about our development program moving forward.
Thank you, Alison. So one of the questions, of course, is we've heard about the wonderful work that Alison and her team have been doing in the mouse model, but mice are not human beings. And the question really is, does this information that we learned from the mice translate into humans? And I think what we're interested in is not simply an improved targeting, and that obviously is good to have, but does it improve the clinical efficacy in humans? So that's really the purpose of the drug. It's not simply to get there. The question is, does it work better, and does it result in a clinical improvement? So one of the ways that we know over time is that there is a difference in health. So one question, of course, is if you can, over time, if we can get the glycogen out sooner, does it result in a better clinical outcome, or do we only get to the same spot faster? So one of the things, of course, is that we know that in animals, this new drug is more potent, so you can use less of it to have the same amount of glycogen clearance. But the question is really not how quickly do you get the glycogen out, but does it make patients better if you remove the glycogen more thoroughly? And, so the question is, how do we prove this? And this is, versus the current standard of care, which is the Myozyme Lumazyme. So in order to the only way that we're going to understand whether this drug works is to actually test it in human beings. So one of the ways that we plan to do that is, in fact, to look at a trial. It's called the Embassy trial, in which we're going to do muscle biopsy assessments in addition to other biomarkers and clinical outcomes, and look in patients who previously have not been treated, and then they will use the standard Lumizyme, Myozyme. And the purpose of this is to really see what do we know about the glycogen clearance, and how does that help us to figure out how that correlates with the clinical outcome. The original LOTS trial, we did not do muscle biopsies in that trial, so we don't know how quickly the glycogen clears. So this particular trial will allow us to see how much glycogen do you need to get out of the muscles, and how does that result in a clinical outcome. So we'll really be looking at samples before and after treatment with recombinant human enzyme. And secondarily, what we'll want to do is really try to understand the burden of the disease in late onset patients. And a lot of this will be looking at various instruments for quality of life, but also exploring new imaging techniques. We'll be doing MRI to look at muscle. One of the interesting things that came up with a lot of the recent MRI studies in Pompe patients is that the muscle becomes identifiably abnormal on MRI long before you can actually demonstrate muscle weakness. So in some of the hamstring muscles in the leg, for instance, the muscle shows evidence of glycogen storage and fibrosis, but you cannot tell that that muscle is weak on clinical examination. So hopefully we can use the MRI to help guide us to determine when do you need to intervene with treatment. And my guess is, and what we're thinking, is that the disease starts probably years before you get a clinical symptom. So when do you need to intervene? And so I think those are some of the questions that we're very interested in. We hope that by doing the biopsies and the biomarkers and the imaging, we'll have a better understanding of the biology of the disease. We'll use that information not only for patients currently who are in treatment with the recombinant rhGAA, but then we can use this information in our development plan to compare the old product versus the new neoGAA, because we're going to have to tell which one is better, and we need to have better sensitive markers, probably more sensitive than a 6-minute walk test, to be able to see if one is better than the next. So just to summarize what we talked about this evening, is we've heard that neoGAA is a biological candidate that we've chosen to take on for further research and development. As Alison mentioned, it's a carbohydrate engineered GAA, and it has a number of different molecules on it, and primarily the multiple bis-mannose-6-phosphate bearing glycans, and we believe that that improves the uptake into muscle, and hopefully it might result in an improved outcome. The neoGAA shows greater potency for glycogen clearance, which correlated in the mice model with functional improvement. And also, the neoGAA appears to be well tolerated in the young Pompe mice. So we want to thank everyone for joining us this late in the evening, and especially to the AMGA and the Pompe community. People who are interested in learning more about Pompe clinical trials can visit www.clinicaltrials.gov. It will have our embassy trial on it and other trials that are currently being done by other companies. And then, as always, if you have any questions that we have not answered this evening, please contact Genzyme Medical Information at the number below. Thanks again very much, and we'll start with the questions that we received on the chat room.
Thank you, Ed, and thank you so much, Alison. That was really wonderful. So, Leo, could you repeat again how someone submits a question online, please?
Sure. Ladies and gentlemen, at this time we'll conduct a question and answer session. In order to do so, you're invited to send chat questions using the chat window to the right of the presentation. Please address your chat questions to the Q&A group.
Thank you. We have received one question so far, which is we received a question about access to therapy in New Zealand. And so I'd just like to say that we recognize that Myozyme is not yet available in every country, and different countries have different regulatory and reimbursement pathways, as you know. So we'd ask you to contact the Genzyme Australia office with your specific request. Information is on genzyme.com, and we will certainly evaluate any opportunities to conduct clinical studies in New Zealand in the future, and I hope that helps answer your question. Another question we have is, and I'm wondering, Ed or Alison, if you could answer this. What is our plan for infants and neoGAA?
Yes. So obviously, we plan to look at neoGAA in infants. The difficulty, of course, is that because the infants are much more fragile and have a very rapidly developing course, what we'd like to do is to make sure in a more stable population, in the late onset population, understand the safety of this new product, understand what the efficacy would be, and then after studying and making sure that it's appropriate for the older patients, then we would look at it also in the infantile population to see if, in fact, it works better than the current formulation of recombinant enzymes. So probably the opposite of what we're doing right now. When we did our initial program for Pompe disease using recombinant, using the Myozyme, Lumazyme, we studied infants first and then studied the adults. And I think with neoGAA, we do the opposite. We look at adults first, make sure it's appropriate, and then study the children.
Thank you, Ed. Another question that we received is, any ideas why you see continued improvement on rotarod in treated Pompe mice versus maintenance of strength by wire hang, which never approaches wild-type levels?
Yeah. It's hard to say exactly why, but I think the one thing is the wire hang test is actually a very stringent test of muscle strength in the Pompe mouse. This is something that potentially could be ultimately a strain difference also between the knockout mouse and the wild-type mouse. I actually don't believe that there was any statistical differences between the wild-type mouse and the 20 mg/kg neoGAA treated mouse in the wire hang experiment. I think that in the rotarod we see an improvement there because we see a decline earlier or an earlier age in the mouse, and therefore we have the opportunity to see that improvement. I hope that answers that question.
Thank you very much. You said another question that we have is, you said neoGAA was well tolerated in the Pompe mouse. Were there any safety findings that were different from GAA?
In the preclinical studies that we've performed to date, we have not seen any safety findings that are anything different from what we've seen with GAA. So far, and to date, in the experiments that we've done or the studies that we've done, we have seen that neoGAA is very well tolerated.
And Ed, maybe you could answer, when are you anticipating the first trials to begin in humans? Can you comment on that?
Yes, the first trials in humans are still a ways off. There is continued research that's necessary in making the product. So from the manufacturing standpoint, we will use the same 4,000-liter GAA that's commercially available, and then it will be modified to add these various glycans on. So that work and research is ongoing. There has to be the preclinical toxicology that needs to demonstrate that it's safe. It has to have a good manufacturing process in place to manufacture it. And then we will also use the data from the embassy trial to help design our studies for GAA in a phase 2 and also in a phase 3. So it is a few years away before we would be actually able to give the first dose of neoGAA, but the work is ongoing, and there certainly is a lot of people at Genzyme that are committed to trying this, and we will be starting the embassy study within the next few months.
Thank you, Ed. Another question that's come in is about a projection of the cost of the 20 milligram per kilogram dose of neoGAA if it gets to market. And it's a very reasonable question, but I think it's way too early to predict what the cost will be. So I wish we could answer that, but it's too soon to say. Anybody? How does antibody response compare between Myozyme and neoGAA?
So I can answer that one. So far in our preclinical work, we have not seen any differences in the antibody titers between Myozyme and neoGAA that are raised in the Pompe mouse model. I don't think that we'll be able to really fully answer that question with respect to Pompe patients until we actually get to the stage of administering this drug to humans.
Thank you, Alison. Another question, these are all really wonderful questions, so clearly everyone was listening very carefully. Does the new compound penetrate the muscle better than current treatment?
We believe that it does, and that's something actually that we're actively investigating at the moment. We have a number of different experiments and different techniques that we can use to specifically look at that. And that really is under investigation. But we believe, based upon our uptake studies in the mouse, in which we've showed that we clearly have much better penetration or uptake into the cell, we believe that that's the case because also, based upon our glycogen clearance experiments, we have shown that at the same dose, we do see significantly more glycogen clearance. So that ultimately tells us that this molecule is penetrating the muscle and getting to the lysosome and clearing glycogen much better.
Thank you, Alison. Another question about Embassy. Is the Embassy study close to commencing? How large a test group and where will it be done?
Yes. So the Embassy trial is very close to beginning. We're hoping, our goal is to try to have the first patient in by April, or certainly before the first half of this year is over. There will be 15 patients in the trial, and we expect that it will take some time to be able to get patients enrolled and to obtain all of the information. It's very likely that we will have more than 15 sites. We're not expecting a lot of sites to see new naive patients, so we're going to open up sites in the United States and in Europe. The team is still analyzing and doing due diligence on the site to make sure that they have the patients and also make sure that they have all the equipment that can conform and they can actually perform the study. So those sites have yet to be determined, but there's a lot of but it should be relatively worldwide, and we're expecting that there should be a fairly good access for patients if they would like to enroll in the study.
Thank you, Ed. Is the stability and enzymatic activity affected by the addition of the glycans?
We do know from our in vitro and analytical studies that yes, the enzymatic activity is affected when we look at it based upon some biochemical measurements. However, what we do believe, and we have some data to support, is that when the enzyme is taken up into the cell, the glycan processed within that cell, and that the enzymatic activity when the enzyme is in the lysosome is similar to that of Myozyme. In terms of stability, we have initiated stability studies. We know that the stability so far looks great. We don't anticipate that we will have an issue with that. We've studied the chemical linkage that we're using to conjugate the glycans onto the enzyme so that that is stable.
Another question is there greater half-life of the neo versus Myozyme?
Actually, in our preclinical experiments, we see that it does appear that the half-life of neo is actually less than that of Myozyme, which again, we think that's a good thing because that suggests to us that the neo molecule has been taking up into the tissues faster. So that's good.
There's a question about Sanofi. Will the Sanofi acquisition affect the program? So I can comment on that, which is that what you're reading in the newspapers and online is pretty much the information that we have. So as soon as we have any additional information about anything, we will be sure to update the community. But you're probably reading and hearing as much as there is to know right now. But thank you for the question. So the embassy study is for Myozyme or neoGAA?
Yeah, and I'm sorry, I should have made that crystal clear. The embassy study will be using Myozyme, the current formulation, and it's going to be used to help design the study when we look at the neoGAA. The eventual study that we'll have to do is, in fact, do a comparison between Myozyme and the neoGAA. So this is a study using Myozyme to understand the endpoints and also to understand the biopsies and how we can use it in the pivotal trial.
Thanks, Ed. Are there any concerns at this point that would suggest that neoGAA would not replace GAA for all patients, Alison or Ed?
From a preclinical point of view, I would say that no, we have not seen any concerns at this moment in time. Ed, I don't know.
Yeah, no, I think it would. It really depends on the data. If neoGAA is in fact better and it's as safe, then I would assume that it would replace the current Myozyme. But obviously, we would know that until we did the study and we determined. And we also would need to make sure that it works in all the populations studied better than what we're currently seeing with Myozyme.
And I believe, I think we've answered all the questions that we have received. And so unless there are any other questions, production issues. Okay. Production issues. So,
I think maybe that question is related to, is there any production issues? And I think we can say at this moment that it's not. As Ed, I think, mentioned earlier, we know that we will be making neoGAA from the 4,000 litre material, and we don't anticipate that we have anything that would become or is an issue with anything related to production at this time. And we can also add that in terms of supply and of the drug, that we are adding additional capacity in our manufacturing facility in Hale with the addition of additional 4,000 litre bioreactors, and that's in process at the moment.
Will the dosing be the same?
Well, as you heard from Alison, the potency appears to be, again in mice, about 5 times better for neoGAA. Obviously, we would have to do the studies in humans to really understand, and it's not so much an issue of the dose, but the question would be, what's the best clinical response? So we would do a dosing study as part of our development, try to determine what is the best clinical response at the lowest dose and use that. So I think right now we wouldn't know exactly what the dose would be. It certainly would not, be more than what we're currently giving, and the expectation that it would likely be less drug, so that it could be a shorter infusion time. But what exactly that dose would be would depend really on the clinical trial results.
Thank you, Ed. So we are coming towards the end of the hour. So if you have any final questions, if you could type them in now, that would be much appreciated. One question is, what studies are you doing to monitor the long-term effects of Myozyme? I don't know.
Well, there are ongoing studies looking at 10-year outcomes in patients. We're also monitoring antibody levels and looking at immunogenicity. And certainly we have the registry, which is one of the best vehicles to really capture the data on patients, and then comparing, the response to Myozyme depending on the age of when the patient started, the condition, various different genetic variabilities, regions of the world. So the registry is a commitment that we have long-term to try to study the outcome measures. And also by doing the embassy study, which again will be done with Myozyme, hopefully we'll learn something that could be used and brought to the current patient population, so we have other tools that we can test how well or any problems with the drug.
Thank you, Ed. Would NEO be produced similar to RH as a powder that is mixed prior to the infusion?
At the moment, we're actually looking at different formulations of what neoGAA might end up in. Our ultimate goal with our formulation development is to make a more convenient formulation. It may be that it's a lyophilized product or a powder that is mixed, but we are also considering a liquid formulation, but that work is ongoing at the moment.
Right, and this question kind of follows up to that, which is there a possibility it could be an oral formulation?
No, there's no possibility it will be oral, unfortunately.
Thank you. Will you correlate embassy histological results with functional clinical improvement?
Yes, actually, that's actually the purpose of the study is to correlate what's happening in the muscle that we see on the biopsy and also on the MRI, correlating that with a functional outcome. So, yeah, that is the purpose of the study. There's also a question again of how many patients would be in the embassy trial, and it would be 15 patients is what we're planning for right now.
Thank you, Ed. And again, if you have any questions about the embassy trial or any other clinical trials, please go to clinicaltrials.gov. And maybe this will be our last question. What functional measures will you look at?
Well, we'll be looking at a number of measures and be looking at health outcome measures. We'll also we will compare it to the 6-minute walk test just so we have an idea of whether or not if there's other how that correlates to other functions. We'll be looking at time function testing and really try to correlate that better in things like a 10-meter walk. So we're looking at a number of different, let's say, simpler functional outcomes that hopefully correlate with the muscle biopsies that are more likely to predict whether or not the drug is working well or how well the patient is doing overall.
Thank you very much, Ed. So I think we're at the top of the hour, and I just wanted to say thank you again to Ed and Alison for giving a presentation here tonight to the Pompe community, but I also wanted to thank Tiffany House and Marcia Zimmerman for initiating this request for us to speak. And again, if you have any questions, please go to clinicaltrials.gov. You can speak to your physician. You can call Genzyme Medical Information at 800-745-4447, and also reach out to the AMDA. So on behalf of all of us here, thank you very much, and have a nice evening.
Thank you.
Thank you.
This concludes today's AMDA webinar. Thank you for participating.